Meiosis is another type of cell division in which the nucleus divides. In meiosis, we are forming sex cells. Sex cells are also known as gametes, and these are going to be egg and sperm found in males and females. Gametes are what we call haploid or N cells. Diploid or 2N cells are cells that contain all of the genetic information for that organism. So gametes only contain half of the information needed to make up the organism, or N. This process can also be called gametogenesis because we're making gametes, and this will happen in the gonads of males and females, and these are male and female sex organs. So spermatogenesis is the formation of sperm, which is made in the testes, and oogenesis is when eggs are made in the ovaries of females. When an egg and a sperm come together and form a fertilized unit, we get what's called a zygote. And a zygote is a fertilized unit that contains all of the information needed for that organism. So when an N egg and an N sperm cell come together, we get a 2N organism, or 2N cell, diploid, which is what you need to be that organism. This entire process of meiosis makes 4 cells. It actually goes through 2 divisions. In males, we will make 4 functional sperm cells, or in females, you'll make 1 egg that can actually be used to form a zygote, and 3 that can't be used, which is what we call polar bodies. So here's a diagram showing how meiosis works. We start with a 2N or diploid cell, and after our first division we get 2 haploid cells. And the second division will give us 4 N cells, all of which are genetically different from each other. These cells right here would represent the 4 functional sperm in males. If this same process were to occur in females, we would get 3 polar bodies, or 3 eggs that can't be used, and 1 functional egg that can be used with one of the sperm to form a zygote. There are 4 steps in the first division of meiosis. The names are just like in mitosis, they just have a 1 or 2 after them to show which division we're in. So for the first division, all the phases will have a 1 after it. So prophase 1. This is kind of like mitosis, in which the nuclear membrane begins to break down. Spindle fibers will begin to appear out of the centrioles, and DNA will start winding into chromosomes. What makes this different from mitosis is we can have what's called a synapsis. This is when homologous chromosomes join to form a tetrad. Tetrad means 4. So like this diagram here, 2 chromosomes come together and they form a tetrad, or 4 chromatids. In this process, we can also have what's called crossing over. This is the exchange of genetic material between homologous chromosomes. They actually take a piece of their chromosome and switch it with each other. Homologous chromosomes are chromosomes that code for the exact same trait or traits. We get 1 from each parent. So in this tetrad here, we could say that this chromosome came from mom and this chromosome came from dad. You get 1 from each parent. Metaphase 1 is when the tetrads, or the 2 homologous chromosomes joined together, line up in the middle of the cell, also known as the equator of the cell. The spindle fibers will attach to the homologous chromosomes at the centromeres. Anaphase 1 is when the homologous chromosomes are going to get pulled apart to opposite ends of the cell. This entire process of getting pulled apart is what we call disjunction. If this doesn't occur properly, then it would be considered non-disjunction. And with non-disjunction can come various genetic conditions like Down syndrome. Telophase 1 is when the cell membrane begins to pinch in to form 2 new cells. Each cell now contains half the genetic material. So in this example, we started with 4 chromosomes, and at the end of meiosis 1, each cell has 2 chromosomes in them. So we went from diploid to haploid. We will then enter meiosis 2, or a second round of division, to further take each of these cells and produce 2 more genetically identical cells, or genetically different cells, that contain half the number of chromosomes. So meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, this process looks exactly like mitosis. So we take an end cell after telophase 1, it'll go through cytokinesis, and each of those will produce a genetically different daughter cell with half the genetic material. Each of the cells produced at the end of mitosis are genetically different from the parent cell, unlike mitosis when they were genetically identical.